Okay, so what we're doing right now is we're setting up what is called a thin layer chromatography plate. So make sure you can come around and you can see. You got to stand over here. You got to actually see it. Because I can put this on the exam and say, what is this? And if you're not watching, you're not going to get that point. All right? So make sure you actually can see what I'm doing. Do not rely on the video. I may decide, you know what? They didn't pay attention. I'm not posting the video. So don't rely on this video, please. All right, so we are about to separate the pigments in these two samples. The first sample is the carrot, okay? So that's one sample we're gonna put on the thin layer chromatography. Again, your lab manual tells you all this information. The next one is the leaf extract. That one is the one that has a mixture of pigments. The carrot doesn't have, it only has one pigment mainly there, right? But we're gonna use it as a control. Now, the other thing is that we're gonna start with, let me have your pencil real quick, Lordson. We're gonna put a line, we're gonna draw a line on the bottom here. It has to be very light. You can't dig into the silica plate because it easily starts breaking apart. That's it, very thin line, like that. Okay, then we're gonna put the sample on the line. We're gonna put the carrot sample and the leaf extract, and then I do it again. I double up, okay? Eventually, we're gonna take the plate and put it in this chamber here. And before we put it in there, we'll put solution. It's a hexane solution. And then we'll put the plate standing up like this. The solution is gonna go through and up the plate. It'll go like this by capillary action. And it will grab onto the pigment and drag it with it. You see that? Now, the interesting thing is this. Where the pigment ends up on here depends on two factors. Factor number one, the pigment's molecular weight. The heavier something is, the slower or the faster it moves?
Slower.
The slower it moves. Good, we've done that before. But it also factors in whatever the pigment's polarity. The interesting thing is this. The silica plate is polar. So if the surface of the silica plate is polar, what do you think it will like? Something that is polar or something that is non-polar?
Polar.
Yeah, polar. Because polar likes polar. Do you remember me saying all that? So if one of the pigments is polar, watch what's going to happen. It's going to interact with the silica plate like this. See? And then when the solution comes from the bottom to try to push it, it'll push it. But it's going to be like really slow. You see? Compared to this one. Another pigment that is somewhat polar, that one is going to bind like this. You see the difference between the two? The one that's polar is binding like this. Very difficult time to get it to pass through the silica or up the silica, right? But the one that is less polar, let's say, will bind like this. Pass the solution through, it'll go like that easily. So after whatever, 10, 15 minutes of running the silica plate, you tell me which band will be close to the top, the less polar or the more polar?
Less polar.
There you go, the less polar, because it'll be easier to push, you see? Which band will be on the bottom?
Polar.
The more polar. But there's also a factor of molecular weight. It's gonna be, where the band ends up is actually a combination of both factors, the molecular weight and the polarity. Sounds good? I focus more on the polarity because I want you to know that part. So again, this is called the thin layer chromatography. And we add the extracts here, we start it, and we watch the pigment go up. So I'm gonna add the pigments right now. We have this capillary tube. I'm gonna open this here. Now it requires us to add it like about, say, 10 times at least or so. I just add it until I get like a dark spot. As dark as possible. You want it as dark as possible, but as small as possible. Again, I'm the one that's setting this up, and we'll get the data and share it together. So here we go. I put one here, and I'll put one here. Okay, anyways, I'm coming back to that. One here, and one here. We do this to let it dry up real quick here. See, the idea is to get the darkest possible, yet the smallest diameter. The way to do it is you let it dry in between applications. Anyways, it's mostly hexane, so it dries really quick. But then you go on the same spot, add another one, same one. It's hard to see in the beginning, but it'll get darker. I'm not catching this one. There it is. Okay. Anyways, we're more focused on the leaf extract. The other one is just a control. How many times did I do it so far?
2.
2 times. All right, 3 times, right?
Is that an alcohol?
I think they put it in hexane too, in the same solution that they have here. I do it twice in case one fails me. I have the other one. If both fails me, I just don't know what to do. I lost track of how many. I think 5, right? All right. I just usually go by the darkness of the.
So the other one doesn't have any color.
Well, the other one has only one pigment, and it's carrot, so it's going to be carotene. But the leaf extract has a mixture of things.
Yeah.
And that's what we're doing. We're going to separate the mixed pigments in the leaf extract using this technique, which is thin layer chromatography.
So it'll end up looking like rainbow?
Not necessarily a rainbow, but it'll have, each band will have its own distinctive color, yeah. But not like a complete rainbow. That's more like separating the light. When you put light through a prism, that's what you get, a rainbow. Rainbow coloration. I'll talk about that, it's coming. Don't steal my thunder. Just kidding. All right. I think it's dark enough, yeah. It's good enough. Good. Now, the solution, the solvent that we use for this is not good for you to breathe. So I actually have one that is already filled. So I'm gonna use this one. This plate is a little difficult to pick up. Okay, here we go. Okay, then I'm gonna put it standing up like this. Then we close it. And we let it run. Okay, it won't take that long. Thank you, sir.